Robot Talk is the podcast that sits down with roboticists from around the world and asks them the questions you always wanted answered. Like, how can robots help scientists? And how does that thing work? Hello everyone! Welcome to Robot Talk. I'm your host, Claire Asher, and this week's episode is about lab automation robots, which are helping advance science by speeding up repetitive laboratory tasks and giving time back to scientists for intellectual work. But first, this is your regular reminder to subscribe to Robot Talk on your favourite podcast provider to make sure you're the first to hear the latest episodes. Another way to find out about new episodes is the email newsletter, which you can sign up to by going to robottalk.org. And if you sign up as a paid supporter on Patreon, you'll get early access to new episodes before they're released publicly. So, without further ado, it's time for me to introduce this week's guest. I recently had a great chat with Yuen Ting Chan from the Natural History Museum about using robots to automate molecular biology experiments. Today, I'm speaking to Yuen Ting Chan, a senior automation scientist and molecular biologist at the Natural History Museum, working on automating laboratory protocols with robotics. Hi Yuen, lovely to have you on Robot Talk.
Thank you, Claire, for having me.
So, when people think of the Natural History Museum, they probably think about fossils and, dinosaurs, and maybe it's not the kind of place that most people would immediately think of there being lab automation robots. So to get us started, could you kind of explain some of the different tasks that robots are being used for at the museum?
So the robots at the museum are there to assist the scientists reduce the time they need to spend on the bench doing the normal repetitive laboratory tasks. Then that gives the researchers time back not to sit in front of the bench and actually get to do other research, maybe analyze more data, work out if they need to process more samples. So I mainly work at the core molecular biology facility. So we are looking at DNA sequencing because a lot of the specimens that's been categorized has always been based on morphology. But like for part of our butterfly collection, they look identical, but then when you do it genetically, you find that they're actually not as identical as you think. So DNA has actually helped us recategorize some of the specimens that we actually have. And the museum is actually involved in some larger projects. So, for example, we've been looking at the bison soil samples. So in the Kent Wildlife, they introduce wild European bisons to help look at how it maintains the woodlands in Kent.
Okay.
And the museum has been part of looking at the soil before and after the bison has been introduced to see the biodiversity and how it's changed over time since it's been introduced. So we've actually been involved in a lot of projects like that, as well as the Darwin Tree of Life. There's approximately 80 million specimens at the museum, which we're part of the project to unlocking the potential of the collection. So we're actually sequencing the actual museum collection that you see on display as well.
Amazing. It's really cool. Can you give some kind of a sense of how long it might take to sequence, a butterfly in the traditional way versus with the robots?
So for the butterfly, it would take like 1 day to extract, and then you've got 2 days maybe to actually do creating the actual DNA sequences library to then load it onto the sequencing instruments. That takes another day. So say about 1 week to process it. While if we use the robotics instruments, we can probably have that library ready in maybe from the extraction all the way to having something ready to go onto the sequencing instrument would take about 1.5, maybe 2 days.
Okay.
And that includes all the quality checks that we usually have to do. So you're cutting it in about half the time that you require compared to someone doing it manually.
Yeah, fantastic. So maybe it's worth, for listeners who might not have, any idea what a lab automation robot looks like, maybe it's worth taking a moment to kind of just describe the kind of machines you're working with and what they actually do.
Yeah, so there's two main instruments that I work with. It's the Tech and Fluent, and the version we have is called a Dream Prep, which is designed for next generation sequencing and preparing libraries to go on one of the sequencers. Okay. And it's got a space to hold all your tips that you need to move your liquids from consumable to consumable, as well as having temperature controlled blocks so that we can do all the steps as required to, for example, the DNA starts off double stranded, but you need to denature it. So we have heat temperature controlled blocks that can cool it as well as have high temperature to denature the DNA. Then we have onboard thermocycler that can do all the steps so that we can multiply our DNA. So we get multiple copies of it, so it actually becomes detectable on the actual sequencing instruments, as well as a fluorometer, which can quantify our DNA and tell us how much we have. So then the Tech and Fluent robot, it can actually perform all the steps from once you've got your DNA extracts out of your organism. It adds the adapters onto it, and then it adds the unique indices that can then means that your DNA strands can then bind onto the sequencing instruments and then give us the actual DNA reads that we need to work out what organism that is. And then at the museum, we also do miniaturization for DNA sequencing. So that uses the SPT Labs mosquito instrument that we own. That one is specifically for pipetting small volumes down to half a microliter. So then when we do the miniaturization of commercial kits where we're using a fifth of the volumes that's recommended by the manufacturer, it can then pipette the half a microliter and the 1 or 2 microliters that we require. So we, for miniaturization, we use a combination of the SPT Lab Mosquito as well as the Tech and Fluent to do all the various steps.
Fantastic, thank you. How hard have you found it to convince scientists at the museum that they can trust the robots to do the job as well as they can, or perhaps even better?
I think this has been a particularly different type of challenge compared to the other fields I've had to set up automation. I think it depends on which stage of the project they've done. So we've had it when PhD students, they've spent 6 months doing the bead-based cleanup manually for 6 months for all their sample types. And when we tell them that it only takes the robot 45 minutes to do that one step, they then realize that they could have saved, it would have meant that all their samples could have been processed in just 2 months, which meant they had 4 extra months to do more work. And they could have contributed more to their PhD. So I think it depends which stage they're at to convince them. So once they've sat through months or years of processing it, they then are a lot more accepting of the idea that if the robot can help them actually move their experiments faster. And then the other issue that a lot of the researchers have at the museum is they're worried about cross-contamination because with their sample types, especially the historical stuff, you can't go back 100 years to collect that sample again. So with the fresher samples, like the soil samples or water, you might have an option, depending on how old the soil is, you can still get more, possibly. So then the cross-contamination is a big thing that we had to show a lot of for our robots at the museum. So we had to think of different ways to do quality checks to show that there isn't cross-contamination within the run as well as between the runs. And then to show that it's pipetting accurately for when we do miniaturization, we even had to weigh the consumables before and after to show the weight changes was correct and like the correct volume had been transferred. So that was using the scientific scale, using 4 decimal places to show that it's actually moving the correct volume across. So we had to do a lot more experiments, and then we even had to design our own single-stranded DNA control because there was no commercially available controls to show that how efficiently our protocols were working. We had to design our own control for the historical DNA pipeline, and then we tried to get as many of previously processed samples so that we could show that even using the automation pipeline still gave you similar results to what you had before. So our aim was generally to show that we can get similar or better results when you use the robotics. We had to produce a lot of data and a lot of repetitive data to show that they can trust and rely it. I think it's more challenging for the historical DNA compared to the more modern or current projects that's is being processed at the museum.
Okay. I guess most of the time you don't have to start your job by convincing everyone that the job you're doing is valid or useful.
Yes, I think at the museum, I think they all prefer doing it manually because they've got more control over it rather than handing it and then we put it onto like the Tech and Fluent and then it just disappears for a few hours and then they come back and it's supposed to be all ready. So it meant that they lose a bit of the control of what is happening in the samples. Are you sure it's happening? But we've done a lot of experiments to show that it's consistent. At least if the robot does get it wrong, it's consistently wrong. And it's not just a one-off. If there was something wrong with the protocol that I wrote, at least it was consistently wrong. And then if it does do it well, it's consistently similar to the results. So we've had to do a lot of experiments to show that how consistent it actually was.
So aside from the kind of human element that we discussed of introducing, robots to the museum's molecular labs, what would you say have been the biggest challenges you've faced so far?
I think for me, it was actually having to write the script for non-standardized protocols on the robots, because usually you would design a protocol and you would have optimized it manually first and it's working well. So now you need to expand it so that you can do hundreds in a week. But that wasn't really the case at the museum. They had their bespoke protocol, which was usually comprised of similar steps to get their DNA sequences. But there'll be small quirks that they've added to it. And then that might just be for that one project that they got funding for. So then to have to write a version of their protocol on the robot so that it would be adaptable for every single project that the museum had. If you think about in any institution, how many researchers you usually have, and each one of them has their own project and their own version of a protocol. It was very difficult to actually make it flexible. But luckily, the instrument that was purchased for the museum, the Tech and Fluent, that was great at making modular scripts where you can mix and match steps. So if there was a repetitive step, you just wrote one script and then you just put it in multiple times. So then by doing that, I had scripts that was optimized and we did all the quality checks to show that it works efficiently. And then depending on what project they had, I just had to mix and match them all and put them together to make a bespoke script and protocol for that project. But it was using all the steps that I could just mix and match. So that made it a lot easier. And also my work partner, Piotr, he was great at getting involved with the research projects at the very beginning when the researchers were getting the funding. So then we could anticipate what projects they were going to do and what kind of work they wanted, as well as having them think the cost for the automation, because the consumables for the automation is a lot more than what you have to use for the manual prepares or any of the manual steps. So we had to also get the researchers to factor in the cost. So he was very good at making sure we were there at the beginning rather than just the final step and asking us, can you do this for me tomorrow? And we have to tell them it's not as quick as that. We need to, first of all, find out what your protocol is. Are you sure it's working efficiently? And then have to make something bespoke for them.
Okay. When you're doing these protocols, does somebody come to you with samples and kind of hand it over to you and then you actually run the robots? Or are you having the researchers actually come in and interact with the robots directly?
So we have the option depending on what they need. So generally, a lot of researchers aren't actually molecular biologists based. So they're usually curators of the museum. So they're very, very good in their field, but not necessarily understand all the laboratory steps required to get the data that they require. So then if they want to, they can just hand over the samples and say, I would like this type of data. So then we would process it all through. And then there's other ones where you have the researchers from a research group where they are molecular biologists, and then I can then train them and get them working efficiently on the robots. So then they log on to the robots and then use the scripts that has been written, because the scripts are all designed so that you choose which one you need, you press play, and then it tells you all the information you require, where you put your plates, and then it'll just work it end to end, and they just have to come back in, depending on if their protocol takes an hour or 6 hours, they just come back at the end to collect their completed samples.
Okay. So what do you think the future holds for lab automation robots, and what kinds of new capabilities would you like to see in the future?
So miniaturization was one of the newer things that a lot more labs were doing, which we're also doing ourselves. And it's particularly useful because we have very low inputs. So then it's nice to be able to concentrate our samples down to small volumes. And then you kind of have more amounts put in for our samples. But one thing that I've seen a lot of in other labs was having a robotic arm that can move your sample plates from one instrument onto another instrument. So then that is something that would have been very useful for us, because particularly at the museum, so most labs already have a pre-amplification labs as well as post. But at the museum, they also have a historical DNA lab, and then they've also got a modern or current or fresh DNA sort of lab. So they don't want to mix historical DNA from like the museum collection with our modern soil, because the sizes of the DNA that we get for historical is a lot smaller. So then it would get lost if it got contaminated with modern DNA. So they actually like to have it as two separate labs. So actually having a robotic arm that I've seen where you can process samples in one lab and then put it through a chute to go to the next lab. Cool. So then it keeps everything more sterile, which is everything that the museum really, really needs, because especially when you're working with historical DNA, the thing that is most dangerous to contaminate it is you, because you're modern and you're current. And if you contaminate your samples, you're going to not get very good results, and there might not be something that you can even go back to. So when, like for the butterfly collection that we've been processing, they take a leg from the butterfly that you see on the collection if you visit the Natural History Museum. Once we've processed it and you've got the DNA, you have to give the leg back and they stick it back on. so then you've got to be really careful how you process the samples and make sure you don't contaminate it as well for the future.
For context, the reason that historical DNA is, I guess, more challenging to work with is because DNA like degrades over time. And so, like you mentioned, it ends up breaking up into smaller and smaller fragments. And so, like, the older the sample, the more challenging it gets. And I know, like, when you start working with really ancient DNA, the lengths that the researchers have to go to in terms of not contaminating their samples is quite extreme.
Yeah, so for the ancient DNA stuff, and even when I used to work in forensics, we had the high sensitivity lab. For cold cases, you had to change out of your clothes into special scrubs, and then you had to wear a coverall that covers your whole all the way up to your head, and then you had to wear hair nets, double gloving, shoe covers, like face masks and everything like that. So if you do shed, you won't shed any of your DNA onto the actual samples that you're trying to process. Yeah, so then I think having an integrated arm that can move plates from one lab to another lab would be very useful for us. And it can also integrate a lot of equipment that we already have. So I think that would be the next stage if we had to process everything.
So I guess that these lab automation robots are not cheap. And so it wouldn't really be sensible to buy one for each of the different labs to keep everything separate. So this robot arm solves that problem.
Yes. So this robot arm, we've seen it where you can have your robotic instruments and other lab equipment in like a special laminar flow hood, so that you have it in boxes, that sterile boxes, and then the arm can just move it in and out of these sterile boxes. So instead of actually having big physical labs where it's like separate rooms, you can have one big lab, but inside there, there'll be like the pre-PCR section would be a perspex, a really big perspex box that can fit everything inside it. And then the arm can move and keep each box is sterile within itself.
Okay. Yeah, that makes sense.
And then for me personally, I've been looking at AI modeling for the optimization of the molecular lab protocols. So because there's a lot of programs out there, but then they need people with experience to feed the model so that they can work out how I would normally optimize a lab process to actually have an AI model be designed to think in similar ways to someone who would be experienced, knowing what temperature would I need to change if I get this result. I think I will change the temperature here or change the incubation period or time, maybe add an extra step here to purify the DNA more if it's contaminated. So like all this knowledge, they need people to feed it into these AI models. So that's something else that I've been looking into, and then seeing how that could then integrate into how you would optimize the automation workflow.
Cool. Yeah, that's really interesting. This makes me think of a long kind of conversation I've been having with various friends and just kind of some roboticists, some not roboticists, about like, what is a robot, right? Like, what is the definition of a robot? Because, you know, we've mentioned some of the lab automation machines that are kind of quite old school now, like the PCR machine, for example. That's not a robot. But the liquid handling robots that you're working with are robots. So, like, in your mind, do you have a definition of what a robot is? It's okay if you don't have an answer. I haven't come up with a good one so far.
Yeah, I suppose if the robot is something that can help you do something automatically, that's why usually we call it liquid handling instruments rather than robot, because it handles your liquid. It's picking up your liquid from one place to another place. I also always see robots as in where they're trying to replicate it, look like an animal, like the dog, and then the robot where it actually walks down the stairs. That seems more like a robot to me than these instruments, which just looks like it's a big box. And then you just have this arm that just moves across and just has these little pipetting tips to move liquid across.
Yeah. I guess the common one is like, is a dishwasher a robot? And I would say no. But I guess the liquid handling robots have more customizability than a dishwasher.
Yeah, I suppose so, because with the washing machine, I suppose that's a machine, so that people don't. It's like when you start distinguishing between a robot and actually a machine.
Yeah.
Because I suppose it has one task to do and it does it well, and you have the buttons to just turn it on and off with all the settings already placed onto the instrument. Because that's how we've written the liquid handling instruments. Is that I've written it all in the background, but when the actual other people in the labs actually have to use the machine, they don't have to do the scripts. They won't see all the scripts and the programming behind. They just press the touch screen and then press play. They put the stuff on and then it just does what it needs to do and they just come back. So it is a robot, but I also feel like it's a very high-tech version of a normal machine that you use.
Yuen, it's been great talking to you today. I've been chatting to Yuen Ting Chan from the Natural History Museum. Thank you.
Thank you for having me on your podcast, Claire. It has been lovely speaking to you. I also want to thank Marie Daniels from UKRES for introducing Piotr and I to you. We always enjoy sharing the work we do at the Natural History Museum. Thank you.
Thanks for listening. If you enjoy Robot Talk, please share the podcast, subscribe, and leave a review. And don't forget to join the Robot Talk community on Patreon if you'd like to support the podcast and get access to some awesome perks, including monthly bonus episodes, behind-the-scenes updates, and more. Visit patreon.com/Claire Asher and join the Robot Talk community today. Next week, I'm speaking to Chad Jenkins from the University of Michigan about robots learning from humans. Until then, I've been Claire Asher, and this has been Robot Talk. Robot Talk Season 6 is sponsored by the Hamlin Centre, Imperial College London.